Hello, I'm Robert Walker, and I'm just going to introduce this paper that I've been working on for some time now, and it's about the timeline for a Mars sample return. So in this video, I'm just going to read the title and the abstract, and I'm going to explain just a little bit about it, but I'm not going to spend much time on that, just to keep this short. So, as you can see from the title, then it says that NASA has to protect Earth. So I'll explain the title. It's almost impossible to get everything into. I can't get everything into the title, even to really understand the background of it. So there'll be some background that some of you won't have. So I have to explain, and I'll do it for the title. So NASA has to protect Earth from Mars samples. So some of you will find that hard to believe, because NASA has been saying it answers the questions during the Perseverance launch, and they said quite clearly there's no life on Mars. And school children asked the NASA experts, is there life on Mars now? And they said no. So why does it have to protect Earth from Mars samples? Well, actually, NASA agrees that they do have to protect Earth from Mars samples. So the things they say may seem not very consistent. Well, actually, it's very similar to back at the time of Apollo. If you'd asked your average scientist, is there life on the moon at the time of Apollo 11, they would say no. If you're just an ordinary person in the public, as it were. But then if you ask, do you have to protect Earth from the possibility of life on the moon, then most of those who are most knowledgeable would say yes, you do. And the reason was because Carl Sagan had published a paper just a few years before in which he showed that, according to their understanding of the moon at the time, that there was a possibility of liquid water just below the surface of the moon, about a few meters below the surface. It was the right temperature. The organics there would be protected from the surface. It could be trapped from the vacuum. There could be liquid water. There could be life, according to the science before Apollo. They also didn't yet know, at that point, they did think that the craters were caused by asteroid impacts. But there was still dialogue about that. There were still people before Apollo who thought they could be volcanic, or some of them could be volcanic. And there was a definite possibility of volcanic processes. That's why when they found that orange glass, they were saying, no, this looks volcanic. And so there could have been things like geothermal vents, and there could have been, and they also didn't know how mobile the dust was. Before the Apollo 11, then they thought that it was a possibility, but before the first landers on the moon, there was a possibility they could have just sunk without any trace into the dust. That didn't happen. But it was only when the astronauts started walking on the moon and we saw the depth of the footprints, and that gave the first clear evidence that the ground there is really quite hard. And it didn't seem to be, and there was a possibility they could have fallen into dust drifts. That didn't happen either. So back at the time of Apollo 11, certainly there was a possibility that there could be life in the dust on the moon, on the lunar surface, protected from UV and cosmic radiation, just being there for a short while and in the darkness, in a shadow. So they did have to protect Earth. And then if you go and look at what actually happened, then they didn't protect Earth. And Carl Sagan was not involved in NASA's proceeding and how they did things. And the way it happened is that NASA, they convened an interagency review panel, basically, but they set it up in such a way that if they had problems with any of the procedures that NASA came up with, there had to be a unanimous consent. It had to change. Since NASA was one of these agencies, that meant NASA could block any suggestion to change their procedures. So the National Academy of Sciences and the National Health Service, they both said that NASA's plans wouldn't protect Earth. But NASA said, and they said that the capsule had to be lifted out of the ocean on a crane onto the carrier. NASA said that they can't do that because the carrier, there's risk of the carrier because it's an aircraft carrier, that it would be travelling, because it's very difficult to manoeuvre them, it could very easily run down the capsule. And they also said that lifting it up onto the boat, then there was a risk of it banging onto the sides and harming the astronauts. So for that reason, they opened the capsule door in the open sea. And the dust from the moon, definitely, some of the dust was very fine dust. It smelled like gunpowder. It would have got into the sea at that point. The worst place to contaminate. That was an executive decision by NASA to override the best scientific advice on the basis that the astronauts' health was far more important. Of course, it is very important, but they didn't even consider delaying the Apollo mission to try and find another solution to this. The deadline was coming, they had no solution, so therefore they just said, okay, we'll just land it in the sea and they'll come out onto a dinghy. You can look this up in When Biospheres Collide, the history of the Apollo planetary protection, of the NASA planetary protection. So that answers, so whatever NASA proposes will get very careful review in case they try and do something like that again. But it's not going to happen like that, because we now have a legal process that didn't exist at the time of Apollo, which was done only under the very weak Outer Space Treaty. The guidelines were, and this means there's no precedent. For instance, the quarantine, there's no precedent for the quarantine. It went through no legal process. It went through no scientific review. Now, so that gets on to explaining the 19 years bit. So if you are familiar with the planetary protection literature, then you may know that the most recent paper on the topic estimated 11 years for the build and 8 to 9 years for the legal process. That would suggest that if they start the build in 2021, they haven't started it yet, but they hope to get to Key Point A in April, and based on that timeline you would think that they could maybe complete it 2031, 2032, something like that. But the legal process would be 8 to 9 years. They estimate it could very well be much longer than that, but you could think that also could be completed. But the difference is, with my paper, is that NASA can't override objections like they did with Apollo, as I just explained. That situation, it will not, I think they just locked in thinking it will be like Apollo. It's not going to be. It's going to be a proper legal process. And NASA can't override objections. And so if something comes up that's equivalent to what they plan to do for Apollo, and they say, oh, the clock is running down, we've got to return by 2031, we've got to start the build, we've already built this facility, and it's already there, we have to use it. That's not going to work anymore. Because it's a proper legal process. They have to show that they're going to protect Earth. And so, for that reason, NASA can't know what they're going to build until the end of the legal process, until all the twists and turns of all the objections have been seen and answered. That's why I say that the 8 to 9 years, and then if you look at the NASA requirements, then they can't actually pass Key Point A. They shouldn't pass Key Point A until they know what they're going to build. Now, it's possible that they manage to convince, it's just NASA talking to NASA basically, it's possible they manage to convince NASA that they do know what they're going to build, and NASA, having the experience of Apollo, may well think, we can override any objections. But when it gets to the legal process, they're not going to be able to override objections. And And the key point B is much more rigorous, and by that point I think they'll realise they've made a mistake in passing key point A, if they do pass key point A. And the reason for this process is that it costs far more to modify a design after you've done it than it costs to build it in the first place. So that's the 19 years. And so the proposal to sterilise samples and return to a stable orbit above GEO. So that's 10 minutes. That's enough for that. Oh yes. No, I just want to say that there's a couple of other quick objections. First of all, you get people saying that it's much more possibility of life on the moon than at the time of Apollo for the moon, but in the case of Mars, although it's only a small number of people who believe that, we have people who are respected who say, but minority view, that Viking already discovered life in the 1970s. We didn't have anyone who thought life had been discovered on the moon. And Curiosity has discovered these brines that are at -70 degrees centigrade, so it's very cold, but they're very multiple lines of evidence, including neutron albedo measurements, that it's there. And at night, it's on the surface 5 to 15 centimeters in the daytime, below the sand dunes that Curiosity drives over. We never had any evidence of water on the moon. So that's another difference. And then there's a possibility, Milton Renner has suggested that a biofilm, and he's an expert in water on the Mars, he runs the REMS weather station on Curiosity, and he has suggested in conversation that that microbes can modify the microhabitats. This is clear that this is very well known, and he suggests that a biofilm could modify this -70 degrees centigrade liquid water into something that is habitable in daytime as it warms up. So that is one of several ways that there could be life in the Jezero crater itself, in the sand dunes. This is perhaps the best known ways, but it's got almost no publicity. And so we definitely have to protect Earth. There's no way that in the legal process there's going to be a change on that. There's no possibility. And then you get people saying, but any life from Mars has got here. Well, this dust and salt has not got to Earth. We don't have any samples of Martian dust or Martian salt. The microbes, the meteorites we have come from 3 meters below the surface in the southern uplands, where the air is very thin. There's almost no possibility of present-day life there. And these were thrown up into space, and there's just no way that the surface rocks even get into space. They don't reach escape velocity, even for quite large impacts. Then the rocks that are actually on the surface don't get into space. It's from deeper below the surface that gets into space. And the dust, and even if the dust and salt somehow could reach escape velocity from the moon, it would just burn up like shooting stars. So there's no way. And then there's the fireball of the entry to Earth and the fireball of exit from the moon. It would burn up just because we'd have to be going fast enough to burn up as shooting stars to leave the Martian atmosphere with escape velocity. So you can't use that argument. And there could have been transfer in the past. The National Research Council in 2009, then it looked at that possibility, and it said that we can't say for sure that none of the mass extinctions on Earth were caused by extraterrestrial life from Mars. So we can't say that it's safe, even if it did turn out to have been transferred in the past. And there's no evidence that life has been transferred from Mars to Earth in the past. So anyway, that's enough introduction. Now that's got a quarter of an hour. Let's see. I'll just read my abstract here now, and I think that's enough. And I'll talk a little bit about the images, and hopefully this will be a bit shorter though than my first try. So the NASA plans, let's bring this up like that. So I'm reading it here because I could put images in it. I didn't want to put images into the abstract for the actual paper, but so I can read it out. So NASA plans to return a sample from Mars by 2031. But however, in 2021 they haven't started the legal process or the build for the sample receiving facility. And as I explained, astrobiologists can't rule out life on Mars in surface microhabits, perhaps relics of an earlier diverse ecosystem. And most of our laws to protect Earth's biosphere didn't exist in 1969, and the Apollo sample return had no legal process, and many lapses of containment, and here I explained all that before. Lunar dust was so fine, and this is when they opened the door, and this is an actual photograph of the divers throwing in the biological containments suit to the biological isolation garment, I think they were called, to the Apollo astronauts, and they then put them on. They then got out onto the dinghy, and then the diver quickly swabbed them down very, very quickly with a solution of bleach. And Buzz Aldrin actually says, as an anecdote, they then threw those swabs into the ocean. So, I mean, really, there was no protection of the ocean. So that's why any NASA proposal will receive careful scrutiny. The legal process can change build requirements. An example, a study by the European Space Foundation in 2012 requires 100% containment of starvation-stressed nanobacteria. This is a photograph. This is a nanobacteria, so they knew about nanobacteria, but they didn't know between this change from the 2009, when the limit was 0.25, to 2012, when with the discovery of these nanobacteria, that they could be starvation limited. They reduced that to a reduction of 0.05 microns. And this is an example for nanobacteria. It's a more recent photograph than this study, and this has just come through one of these nanopores, which is only 100 nanometers in diameter. And for this reason, they actually said that, and this is beyond the range of a HEPA or ALPA filter, they aren't even tested down to 0.05 microns. And if you look at the study, it's an actual requirement that no particle of this size should be released through the entire lifetime of the facility under any circumstances. Containment. And this is beyond the reach of HEPA and ALPA filters and may become a legal requirement. After all, SIN, the most recent European Space Foundation sample return study. This technology doesn't exist yet. And in the paper itself, I look at the current technology, which is the experimental filter, which is not available commercially, only in the labs, tested in labs, is able to contain 88% of individual SARS-CoV-2 viruses, or water droplets small enough to contain an individual virus, and that's 60 nanometers. It's just a bit bigger than this requirement, and it can contain it at 50 nanometers. And that means 12 out of 100 will get through, and the requirement is that none out of 100 can get through. So the technology doesn't yet exist, even as experimental filters in a lab. NASA can't start the build until they know if their proposed facility will be accepted or modified, and they obviously can't know that yet. They haven't even the specifications and studies for Mars receiving facilities that I've seen in quite a few papers suggesting different ways of doing it. They all have technicians handling the samples. They all use HEPA and ALPA filters. None of those will be accepted if this is the legal requirement. Then none of those will pass legal review. So NASA can't start the build till they know if their proposed facility will be accepted. So adding the timeline for the build to the legal process, we find that the facility can't receive sterilized samples from NASA before 2040, I should also mention that this also applies to the ESA launch of the ESA sample fetch rover and the orbiter and the capsule sent up to orbit. All that needs legal review because they have to break the chain of requirement from Mars to Earth, and it's entirely possible that what they propose is not accepted. So that would suggest you can't launch any of those until 2019, 18 at the earliest. And it's actually very unlikely the legal process is completed by then. So we examine, and so I make it 2040 as the first year that it could return, but this is very optimistic, as very optimistic projection. And we examine one solution is to sterilize the samples sufficiently for planetary protection requirements while preserving both astrobiological and geological interest. And this is the actual data I found was for gamma ray sterilization using cobalt-60 or something like that. And the level that would sterilize any life from it, even something 10 times more hardy than radiodurans, could be sterilized, and even actually much more than that, even a higher level, it would still be recognizable as life. But it would obviously not be alive, and then this would not have any effect on the geology, at least with gamma rays. The only effect would be that salt would turn blue and quartz would turn brown in colour, but it would still be recognizable what it is. It wouldn't change the distance between the planes of the crystals. It wouldn't change the radioisotope dating, radiometric dating. That would be exactly the same. And basically there's no problem with doing the geology with this level of stabilization. And then I suggest using an X-ray, because this could be a very low cost way of doing it. And again, if you could build this into the ESA sample return Earth entry vehicle, then just maybe build in, because they plan aero capture from Mars, and just directly launch from Mars orbit and aero capture into Earth's atmosphere. And that's why I said that will definitely need legal scrutiny and careful scrutiny as to whether that is an acceptable way to return it, as well as the way that the sample is transferred to that from the Mars Ascent vehicle, and many other details would need very careful legal review. And so the simple way is just to sterilize it. So I suggest these nanoscale X-ray emitters. So that's one way of doing it. This used just amps rather than kilowatts. Just watts rather than kilowatts. Amps as well, sorry, but also watts. Anyway, this makes it an unrestricted sample return, a relatively simple process. There wouldn't be any legal process to speak of, just the normal one that you already have for return from a comet to an asteroid. However, there may be a significant possibility of viable present-day life. And if you think the Viking discovered life, for instance, or if they discover something new that really suggests it. If so, we propose NASA return the sample to a holding satellite in an inclined, that should be inclined, not inlined, orbit above GEO in Earth's ring plane. Oh, I've got a much better image for that. Let me just bring that up. So we have to bring down a bit. There we are, equatorial orbit. Then I can give you a much better image for that, which I've done since. I have to copy that over. Bring it down a little bit. There we are. And let's bring that up full screen. So that's the idea. We turn to an Earth's ring plane, that past plane above geostationary Earth orbit. It's where Earth would have a ring system if it did have rings. It's inclined at 7 degrees to the equatorial plane, and the orbits there don't drift. And it turns out that if you put something, even if you had an explosion of the sample receiving facility, so this is my suggestion, that I don't think anyone suggested that before. So it's inclined at 7 degrees to the equatorial plane. It's been suggested as a disposal orbit for geostationary Earth orbits, satellites, and so would be much higher than the disposal, say 50,000 kilometers from above Earth's surface. So at that distance, instead of that distance, very far from GEO, low latency from Earth, then that would be a place where you have, as far as possible, the furthest you can get in delta V from Earth and from the Moon, and also it naturally contains any debris as a result of any off-nominal events, or even just kind of breaking off insulation. So I suggest that as a place to return it to. And so I find it has many advantages for protection of Earth, the Moon and other satellites. And then sterilized subsamples can be returned to Earth immediately, so there's no delay for geological studies. And then if the unsterilized samples are of astrobiological interest, they can study them with low latency from Earth, teleoperating instruments such as the ones astrobiologists design to search for life in situ on Mars. And we have just marvellous instruments we could send to Mars, and that they've designed, some that could detect even a single amino acid. Now, this article examines specific worst-case scenarios, such as a blue-green algae with everything flipped, as in a mirror. And so I've got this diagram here. So DNA spirals the other way. Let's just bring that up. Normal life, then mirror life, just flipped, as in a mirror. And this is to give the idea that you could get algal plumes of mirror blue-green algae in the Great Lakes, say, or in the sea. And it could be of mirror life, as a result of returning a blue-green algae that's flipped in a mirror from Mars. And the problem with that is that most normal life can't eat mirror organics. There are a few microbes from Antarctica that can, but most life can't. And that's the DNA, amino acids, sugars, fats, and none of that is accessible to normal life. But Martian mirror life, there's lots of mirror organics. Indeed, most of the surface and normal organics, both chiralities, most of the organics on the surface of Mars is of both chiralities, because most of it's from comets or from meteorite impacts. And there might, of course, also be the normal life as well as mirror life on Mars. Indeed, if you think about panspermia, it could even be that for some reason the normal life got here and the mirror life didn't. We don't know why there isn't mirror life on Earth. And synthetic biologists have started the process to gradually turn normal life to mirror life, and they just change one bit, a bit, and then another bit, and then another bit. And to change the whole thing, you can't just flip it in a mirror. Change the whole thing, then that will take probably years to decades, but they can see the kind of route plan to get there. And they warn that if released from the lab, such life would gradually turn organics in terrestrial ecosystems to indigestible mirror organics, giving it a competitive edge with normal non-mirror life. And here I assume that the readers already are familiar, and I talk about that in my article, about more mundane things like life from like opportunistic pathogens like botulism and like tetanus, and then Legionnaire's disease is an opportunistic pathogen. None of these are adapted to humans. Legionnaire's disease is a pathogen of biofilms that infects the lungs using the same method it uses to infect biofilms. It's not adapted to humans, and it has killed humans. And then immunocompromised people can die of fungal infections. And for most of us, if you got it, you get a fungal infection in your nails or something, but you can actually die of that if you're immunocompromised. And then one of the microbes that pass the tests for being best able to survive in the very low conditions, low atmospheric pressures on Mars, is one of these. It's a microbe that is a problem in hospitals for transplant patients. It also causes all sorts of problems. I can talk about that in a bit, and with blood fusion and so on. And the problem is that if it is unrelated to terrestrial life, then we might essentially have a compromised immune system. That our life has never encountered something like that. And if you assume that the immune system develops in response to threats that evolutionary Earth life has encountered in the past, then it is entirely possible that we have basically a compromised immune system. In particular, for instance, the antibiotics that work with cells walls of microbes, and these are the first line of defense. They probably do nothing to alien life. So there are lots of issues, but Milver life is one of the most is one that really brings up these issues really into sharp focus. Synthetic biologists have already made, and this is Hachimoji DNA, which has got 8 bases instead of 4. And these 8 bases, the extra Hachimoji DNA or Hachimoji RNA, then they add 4 different bases and different ones. And so how do the synthetic, and they have actually made that in the laboratory. It's inheritable, and it expresses an aptamer that actually acts as an enzyme that can make spinach glow green. So they show that it worked. And the synthetic biologists, after all, we get occasional leaks from the most secure laboratories, and these are not considered safe enough for synthetic life. And keep Earth safe by designing synthetic life to be dependent on chemicals only available in the laboratory. We can't do that with Mars sample return. And then if you go, do I talk about quarantine? I don't think I talk about quarantine here. I'll just quickly mention quarantine, and I'll talk about the last results. So this is obviously, if that is what happens, then there is just no way that the unsterilized sample return can pass legal review, because we don't have anything like technology that could contain river life. We just don't have it, and quarantine can't do that. And there are many major issues with quarantine. It never passed legal review, it never passed scientific review. And remember, people from many different disciplines are going to be looking at this, an open process that synthetic biologists will be looking, the epidemiologists will look. And this word is now familiar to most of us, but it wasn't before. The first thing epidemiologists are going to ask, one of the first things, is how do you quarantine an asymptomatic carrier, a lifelong asymptomatic carrier of a human pathogen, like typhoid Mary, or a symptomless supercarrier. Super spreader is the word that's more familiar to us since COVID. And the answer is no way to do it, as far as I can tell. So quarantine just doesn't work. And it never worked anyway, really, because they, as Carl Sagan pointed out, they didn't know the length of the quarantine period. There's no way to tell if it's an unknown pathogen. For leprosy, it's decades. And then the question was, the ethical question, what do you do if a technician... So the reason we need quarantine for Mars sample return is if technicians are handling the samples directly, and then it's a cut in the glove and they get infected, as happened with Apollo 11. I think it was the case even for Apollo 12. I think it was the case for Apollo 11, just one technician then. 11, I think, for Apollo 12. I'm not sure of those numbers. I've got it in the paper, so you can check. But anyway, and with the sites and everything. So quarantine would not protect against this. And then you've got the problem, like if the the capsule, if there's an accident, retrieving the capsule and taking it to the sample return facility, or if you get a plane crashes into the sample return facility. There could be many ways in which you get breaches of containment, and then you might need to quarantine. But quarantine won't protect Earth, not from mirror life. So if mirror life, you get a fungus of mirror life that gets onto your skin and it doesn't do very much, you hardly notice it. And a good example of that is actually with the ISS. Their zinnia, the flowering plants they grew there, edible flowers, and it suddenly wilted and died. And the reason was because of a fungal infection that one of the astronauts brought there. And it was actually an opportunistic pathogen of humans. A fungus that can in some humans, especially in hospitals, be an opportunistic pathogen. But it didn't do anything to that particular astronaut. But it is also a pathogen of plants. And this fungus just killed their biology experiment. And so that's a clear example of how you could have, if you had a similar opportunistic pathogen of, say, Martian astronauts that got into their skin, they're not even aware it's there, it wasn't visible fungus, this astronaut had no idea that he was carrying a fungal infection and destroyed that plant. So how can you keep mirror life out of Earth? The answer is no, you just can't do it. A quarantine can't do that. So, and so as I say there, in some worst case scenarios studied here, Mars has life forms that can never mix safely with other life. I'm just checking the time, I think I managed it. Other life in Earth's biosphere, and quarantine of astronauts can't keep it out. And so the future possibilities, and so that's for Mars as well, astronauts on Mars as well. I should say technicians there, but I also mean astronauts. The future possibilities and opportunities open to us depend on whether there is life on Mars and what its nature is. Oh yes, I think I do mean astronauts. Finding the answers seems a top priority for both space colonization enthusiasts and for astrobiologists. And with a combined effort, astronauts could conduct rapid surveys of the most important potential habitats from orbit around. They could do it from orbit around Mars by telepresence. And humans would be important because of the minimal latency and the decision making we can bring to this situation. And basically, it's a really exciting orbit, the one about, it's like astronauts like being in ISS and looking at the Earth turning below. This is a spectacular orbit that comes close above the two lunar poles and then very close to Earth twice a day. And telepresence orbit of the HERO mission, that's a proposal for a way to do this. And then they go into they would do avatar, and they would use just like playing in a game, like virtual reality in a game, and with goggles and everything. And they'd be basically on the surface of Mars as if they were there. They would actually experience Mars more directly than in a spacesuit. It would be a very exciting mission to be exploring on Mars, looking for life from orbit. And you can jump from one avatar on the surface to another. There'd be many, if you do this properly, you would have hundreds of landed robots. And so with a combined effort, and this could help resolve the answers question quickly, and both Viking landers made observations that are hard to explain as non-life, especially the offset of 2 hours, and so I'll just explain that. So the red here shows the temperature fluctuations, and this is the Viking labelled release, where they fed radioactive carbon, they mixed radioactive carbon organics amino acids with the sample, and with a sample scooped up from the Viking surface in 2 different boat landers did it, they both got the same results in widely different parts of the moon, where Viking 2 was much further north and much colder, and they both got the same reaction. And what they did, they fed the radioactive carbon, and it was quite a simple conceptually, that they then just measured the gases evolved, They don't know if it's carbon dioxide or methane. This was back in the 1970s, and instruments weren't miniaturized like they are now. And they just measured these evolved gases. And Joseph Miller, he discovered that decades later. He just suspected this from looking at the data, and they had to do a very long, they had to actually recover the visual observations, which weren't publicly available, and it was really quite a big, long thing, actually recovering the data, which was they didn't even know the format originally, and then they had to, I think, had to actually deal with paper observations that one of the team people had, paper printouts. And that was a real adventure. That's a very interesting adventure, the way they actually got this data here that we can now look at. And he discovered that because it just wasn't available to do this proper analysis until they did that retrieving of the data that hadn't been looked at so carefully. And he discovered that these are the evolved gases, and to his eyes, he sees that the peak is much, much later than the peak from the, and it's offset by 2 hours, he says, and it doesn't follow exactly the peaks of the temperature. And you expect an almost exactly the same double peak, not only offset by about 10 minutes, if it was a chemical reaction. So to his eye, it suggests circadian rhythms. So we need to understand those results. So there is a chemical explanation and there's a life explanation. Neither of those are 100% satisfactory. The major problems with the chemical explanation, for instance, why would the chemistry be inactivated when the dust that came from the Earth's surface was kept in darkness for several months before it was added? That's quite difficult to explain in terms of chemistry. And there's several other anomalies that are quite difficult with chemistry, including the circadian rhythms. There are also difficulties with the life explanation too. And so we need to understand these results, either as life, unusual form of life that we don't yet understand, or as complex chemistry. And the reason they didn't have they were confused by the perchlorates. And for that reason, this paper suggests modifications to the ESA-FET rover to add a sample of dirt additional to the rock samples. The dirt might even contain present-day life if it is very abundant on Earth, on Mars. And for instance, the oxygen emission that Curiosity discovered, the oxygen levels anomalies, if that was actually produced by life, and it's not, they can't rule that out. If that was actually produced by life, it would actually be quite abundant there. It would actually be more abundant than life in the Atacama Desert, say, or the McMurdo Dry Valleys. I think all getting on for. I did an estimate, and it's not quite quite so much emissions as a biocrust, but it's much more than an endolith or life living on the surface of rocks, and it is not beyond the bounds of possibility if these brines are indeed inhabited. We don't have brines like those, not the exact analogies, in the Atacama Desert or the McMurdo Dry Valleys. It's not impossible that the oxygen anomalous readings that Curiosity has discovered could be caused by life. If it is, then it could be quite abundant, and in any case, if the 2 Viking missions did discover life, then you'd expect to find life there in the dust, because after all, as long as you've got a big scoop, you've got a scoop similar to the Viking lander, and there's the Viking scoop, and if you could just make a scoop like that for the ESA-FET rover, that's a mock-up of the ESA-FET rover. This is just CGI, it's not the real thing. It's just quite good CGI. So if you could just put that on top of the sample return capsule, then that sample of dirt would be of great interest. And the Perseverance is going to make it's going to take probably 1 regolith sample. And being geologists, they're probably going to look at the rocky areas I'm not sure that they're going to even return some of this dirt. And so it would be a very small sample. And so I just proposed that the ESA Fett rover and the capsule, this container hasn't been sent to Mars yet. It was going to originally, but now they're just depositing the sample tubes on the surface. And the ESA Fett rover is going to pick them up like that. There'll just be tubes on the surface, and it's taking the capsule there. So the capsule, hasn't been made yet. So we can modify it. We could, for instance, have a plate on top to put the dirt on. Another possibility I suggest is putting magnets, taking an extra couple of these sample tubes, putting magnets just inside and below the top of it. And leave that vertically on the surface, if they can manage to stick it in the dust vertically. Or attach them just vertically to the rover, where they sit on the rover while it's driving around. I just thought of that just speaking right now. You could have a couple of the sample tubes fastened vertically on the rover, and it just collects dust, and the magnet is on the rover. And that could happen as well. And that attracts the dust. And then, so one thing I worked out in my paper is that if the biofilms produce little propagules, and then the dust storms send things of 0.6 millimeters, that's the size of the grains, then they don't get suspended in the atmosphere, but they do great big saltation leaps, and they can go from 10 to 100 meters in one leap in the dust storms, and they bounce and bounce and keep going. And that we quite likely think for life to exploit. And the biofilms, they could cover themselves with nanoparticles of iron oxides. And I worked out that a 0.6 diameter could contain 40 million microbes at 0.1 microns with hexagonal close packing. So I think it's 41 million. So there seems a distinct possibility that somewhere in that dirt or in the dust. So why not collect the dust? And it's very interesting to know what the dust is. Whether it is, some people say the dust is so because of the perchlorates turned into chlorates and chlorides and the UV radiation, that this was just sterilized terrestrial life. So we need to know about that for forward contamination, whether it does or not. And then it might just possibly have Mars life that's adapted to it, and maybe at particular seasons. So that's another suggestion. And I think if we do all that, then we could make it a much more interesting sample for astrobiologists. Basically, it's sadly the sample tubes that they sent to Mars, they have a probability of 1 in a million of viable Mars organism per sample. The design criterion is 1 in 1,000, they achieve 1 in a million. They can have up to 1 nanogram of DNA per sample tube, per gram of sample tube. And so I think it comes up to something like 150 nanograms you could have per tube of DNA, or you could have that much of amino acids. You could have 8 times that amount of total organics. So these are not sterile tubes. Although they are the most sterile things we've sent there, they're not sterile in a sense that astrobiologists would hope for. And so we need to bear that in mind, and that if we were to send the in situ instruments to Mars, they detected just a single amino acid, then that is something that can be of significance. But coming back from Mars, unless it's mirror DNA or something, then it would have to be quite a lot of it for us to detect it as life. And basically Perseverance, they don't think like astrobiologists. It's basically a mission designed by geologists, and they have designed it on the basis that they think that if they can return something that has similar level of organics to the Martian meteorites we have already, and if they can tell the astrobiologists, and so we have these Martian meteorites that have multiple biosignatures, and the astrobiologists don't know if it's life or not. And the geologists think, well, if we just can tell them where it came from on Mars, it'll answer all their questions. And that's just not how it works with astrobiology. they don't know if it's life. And returning an exact duplicate of the Tissin meteorite, say, from Mars, and you know better where it came from on Mars, is not going to resolve the question as to whether those are biosignatures or it's life. So it's not likely that the past life, because of these high levels of contamination, first regards as to biology, then unless it is a very clear signal, but it's likely to be highly degraded. But I did come up with some ideas in my paper about there's a possibility of newly formed craters. And I worked out that you could have a crater 16 to 32 meters that was excavated in the last 50,000 years, which is very young. And that there's probably such a crater within Axis of Perseverance. And that would have excavated to a depth of quite a few meters. And then there could be, there would be very small ones that even there's a chance of a small crater is excavated in the 4 years or 10 years or 4 years. But that would be very small, but it could still be half a meter of excavation. It could be useful. And so I have some discussion about ways in which you might be able to do that. Indeed, you might be able to use the Mars copter, but I don't know if they would consider such a thing. But anyway, so there's no other terrestrial life planet within reach to study the chemical processes that develop on a planet left for billions of years without life. If the apparent circadian rhythms are due to complex chemistry, that's possibly of great interest. So this is the idea that Mars is of great interest even if there's no life there. And it could easily miss present-day life, but it can be a useful first step to inform those future searches. And for instance, if we find out more about the Viking and what their experiments actually meant, then that would be a great step forward, because we would then know what not to look for if it isn't life, and understand better the chemistry there that would confuse the searches for present-day life. So this article concludes that the complex laws already in place to protect Earth's biosphere are both understandable and necessary. So anyway, that is my presentation, and I'm going to upload this. I'll give it a little bit of thought. Maybe this is the one that I will share with some of you, but we'll see what it looks like. And so just in case it is, my aim is to try and get feedback. And so just to make absolutely clear, I'll put a link in the video description. That is my article, and so you can go and read about all this. It's got all the citations and everything, and so I'll give a site in the video description to the article. That should bring up that bit. So that's the article, and you can download it and read it. And I'm very keen to have feedback. The main thing I produce, I'm not an expert on any of these things, and I am multidisciplinary. My training is in maths, but I was encouraged by an astrobiologist friend to say that I should try getting this published. And my aim right now, it is a preprint. It's had no peer review, and I would like to get feedback from the experts. So it's multidisciplinary. The main thing I've done, and the main reason that it's got new things in it, is because I have been very multidisciplinary. Like, nobody's looked at synthetic biology before and its connection, or epidemiology, or the engineering of filters. And these are things that would definitely come up in the legal review. So I'm just anticipating what has come up in the legal review by rather broadening the literature beyond the usual point at which people have been looking when they have worked on the previous planetary protection papers. And as a result of that, of course, I'm touching on many different disciplines, and I've been as careful as I can be, and I've read the papers carefully, and you can double check them. So I'm going to be, I think when I ask the people whose figures I've got haven't yet got permission for the figures for the article, so that's my next thing to do. And I think when I do that, then I will also ask for comments from them. And I'm going to share this, probably share this with the Virtual Astrobiology Facebook group, with my astrobiologist friends, and with anyone, and people in other disciplines, and engineers and so on. And just see if anyone has, if there's anything I've missed, anything I haven't understood, any confusion, any slight confusion, even the typos, but whatever it is, do please let me know. And please, anyone who's watching this is not peer reviewed. So please, I say that right at the head of the article. I say that, most preprints don't say that for some reason. If we go up to there, it says there right at the top. Please note this article is not yet peer reviewed. It is currently in process of development with frequent updates. So please bear that in mind. It's not published, it's not peer reviewed, it's a preprint.